اوكي، ان شاء الله يشتغل، ان شاء الله يشتغل. اخر مره سويت 2 ريكوردينجز وما ضبط. حمدا لبارئ الانام، ثم الصلاه والسلام ما ناح في دوح الحمام على الرسول العربي واله وصحبه ومن تلا من حزبه سبيله في حبه على ممر الحقب. اللهم إنا نسأل زلات فاغفر زلات ألسنتنا، وللكلام سقطات فاغفر سقطات كلامنا، وأنت المثبت فثبت قلوبنا على الطاعة واعصمها من المعصية، واجعل لنا في سائر أمورنا خيرا. Today, very easy lesson. Very easy lesson. Okay? Oxidative phosphorylation. There will be some things that you will need to memorize, but overall, inshallah, very, very, very easy lesson. Let me move this so you can see me better. طيب. Objectives. Okay, you know, aerobic respiration versus glycolysis, right? Or acetyl-CoA production. But first, it's always glycolysis, then link reaction, where you produce acetyl-CoA, and then you'll have TCA cycle, Krebs cycle, and then you'll have oxidative phosphorylation, where most of the ATP will be produced in aerobic respiration. Now, first you need to know what is oxidation and what is reduction. Reduction, gain of electron. Oxidation, loss of electron. But in biology, reduction, you gain an electron and a hydrogen with it. Oxidation, you lose an electron with a hydrogen with it. Okay, an example, you see NAD+, that's oxidized state. Malate plus NAD+, oxaloacetate plus NADH. So, okay, NADH is reduced. Now, you know, in biology, it's always redox reaction. If there's something reduced, there will always the other molecule will be oxidized. So it's always redox reactions. Okay, I said that NADH got reduced. That means I will induce that malate got oxidized from malate into oxaloacetate. So NADH gained an electron and hydrogen, while malate lost an electron and hydrogen. What are NAD and FAD? You saw them a lot in Krebs cycle, in glycolysis, in link reaction. What are they? They are electron carriers. They are your delivery man. They are مندوب التوصيل. حق hunger station, Kita, delivery. Okay? Delivery guy. But rather than delivering food, they deliver electrons. First, they get reduced. Either in the Krebs cycle or in the link reaction, and then they get oxidized and release the electron they took from there into the oxidative phosphorylation. So they take the electron from there and release it there. Electron carriers, because they can be oxidized and they can be reduced. Electron transport chain. I will explain everything in the next slide. One picture, I will explain everything. Okay? You will need to memorize this sequence, but it's going to be very easy. Okay? And from there, you will understand everything, even the oxygen, like, you'll understand everything, inshallah. طيب, let's start with NADH. We said that NADH gets oxidized, okay? That means it releases an electron and the proton, which is, or hydrogen, the proton. Hydrogen ion, proton, I will use interchangeably. So when I say proton, I mean hydrogen ion, because it's more easier to the tongue and it's faster. Okay, we have NADH, it will get oxidized. Now, its electron will go to where it's going to get oxidized at a protein called complex 1. What's another name for that complex 1? NADH dehydrogenase. Common sense. It will oxidize it. It will take its hydrogen, dehydrogenase. It will take its electron and it will release its hydrogen as a hydrogen ion, proton. Okay. Now, what will it do with the electron? It will pump it. It will pump it through the inner mitochondrial membrane. You need to realize that mitochondria has two membranes, outer and inner. Outer, we don't care about it, but we care about the space between the outer and inner, which is called inner mitochondrial membrane. Inner membrane space. And you have the inner mitochondrial membrane, where all the proteins and everything that we're going to talk about is in. So it's all, the proteins are all in the inner mitochondrial membrane. Complex 1, what is it? Inner mitochondrial membrane. What's another name for complex 1? NADH dehydrogenase. Now tell me, Al-Hassan. Okay, I've oxidized the NADH. You've pumped the electron. What does that do? Pumping the electron will produce energy. Okay, produce energy then. It will produce the energy, and that energy will be used to pump the proton out from the matrix. A proton is H plus ion. Pump the proton out into the space between the two membranes, in an intermembrane space. I don't know what's its name. I forgot, to be honest. Okay, where does that electron go? It will go to a molecule called coenzyme Q. Now, the coenzyme Q will not pump a proton, will not do anything. It will just take that electron and move it to complex 3. So 1, coenzyme Q, 3. What does 3 do? Pump the electron, and that will produce energy, actively transport a proton into the inner intermembrane space. Okay, now same thing. Now the electron will be pumped to where? To cytochrome C. So can you see the electron will not be pumped from one complex to another immediately. There will always be something in between. First, between 1 and 3, you have Q, and now from 2, from 3, it will go to cytochrome C, and the cytochrome C will go and will take the electron into complex 4. So 1 Q, 3 C, cytochrome C, into complex 4. Complex 4 will do the same thing. It will pump the electron and will actively transport a hydrogen ion. Now, what's another name for complex 4? It's cytochrome C oxidase, because it oxidizes cytochrome C and takes that electron from it. Okay, you've been telling us, okay, pumping protons, what does that do? Now, I'm actively transporting H+. I'm creating a high concentration at the intermembrane space. Now, I'm creating what's called an electrochemical gradient. I'm creating a concentration gradient, basically. Now, H+ want to go back to the low concentration inside the matrix, inside the mitochondria. But it cannot move through the phospholipid bilayer by itself. It needs something to let it through. What's that? The ATP synthase. Now, H+, it will still move, right? It's passive movement, concentration gradient, but still moving. It has kinetic energy, okay? And okay, what does that mean? The ATP synthase, because H+ will move through it back into the matrix. The kinetic energy of the H+ moving through the ATP synthase, the ATP synthase will use that energy, will use that kinetic energy, and utilize it for coupling ADP and inorganic phosphate, creating ATP. So to use the kinetic energy to phosphorylate ADP, producing ATP, voila, you will have ATP. Now, where is oxygen on all this? Oxygen, oxygen, oxygen. Where is oxygen? It's in the last step. Last step. So, okay, I have the electron pumped, everything. I have the hydrogen ions going back. Who's going to accept them? Because if then they'll go back to the NADH. Okay, I'm not having NAD plus anymore. If I don't have NAD plus anymore, I don't have oxidized state of NAD and FAD. Big problem, because then the link reaction cannot proceed, Krebs cycle cannot proceed, all of aerobic respiration cannot proceed. So who takes them? Who takes them away and let NAD and FAD stay in their oxidized state? Oxygen. Oxygen is the final electron acceptor and proton acceptor, but mainly it's named final electron acceptor. It accepts 2 electrons, 2 protons, 2 H+, and produce water. That's the only importance of oxygen, but it's one of the most important steps, because without it, it's just a domino effect. And that's basically for NADH. Now, okay, you talked about NADH. What about FAD? Same exact thing, but rather than complex 1, it goes to another complex called complex 2. It's gonna be between those. And what's the major difference between complex 1 complex 2? Complex 2 will oxidize FADH and will take its electrons, but it will not pump a proton up. So it only move the electron into the coenzyme Q, and then the oxidative phosphorylation will just happen same as normal. And that will be important later for when we say about ATP production per NADH and FADH. Now you just need to memorize complex 1 for like NADH complex 1, coenzyme Q, complex 3, cytochrome C, complex 4, and then the electrons will go to O2. For FADH, the same thing, but rather than complex 1, you'll have complex 2. Now, this is the major ATP production because this produces at least 25 to 27, around that number per cycle, right? In Krebs cycle, you only have 2 or 3 ATP produced. In glycolysis, you have only 2 net ATP produced. So see, that's the biggest difference. It produces almost 90% of all ATP. Cytochrome, you just need to know what is a cytochrome. It's like a type of molecule with a heme group, propanol ring. You just need to know, like, I think the cytochrome P450. It's a molecule that's going to be used when you metabolize drugs, medicines in your liver. So, like, these important metabolism, you'll have cytochrome P450 used there. What type of cytochrome you have in oxidative phosphorylation? Cytochrome C. Going to, okay, coenzyme Q10, you just need to know that some studies has said that, okay, maybe it reduces muscle pain with people that are taking statins, which are drugs that are used to lower your bad cholesterol and cholesterol overall. So just know, but it's not like proof, but it has been like there are some links with coenzyme Q supplements and lower pain for people that take statins. This is basically what I all said. Everything in that image, everything I've said there, moving through the ATP synthase and the kinetic energy, everything that I've said. Okay, I told you that it's important about the FADH and how that complex 2 does not pump a proton up. Why? Because there is a difference of energy. NADH, because it have 3 complexes that pump proton, it will generate 3 ATP. But FAD, even though the electron is pumped in 3 complexes, but only 2 complexes will pump protons. So only 2 ATP will be produced per FADH. Now, it's not something that, oh, because 3 complexes, that means 3 ATP. No, it's an approximation, but it's just for you to memorize, right? 2 ATP, 2 complexes, 3 ATP, 3 complexes. And then you will know if it's NADH or FADH2. Going there, we have inhibition of electron transport chain. If I have inhibition of electron transport chain, I will not have ATP, less synthesis of ATP. Oxygen will not be used, so less use of oxygen consumption. And NADH will not be oxidized. So oxidation of NADH will reduce. And now, what are the inhibitors of NATAC? You need to know 4. You need to know rotenone, which binds to complex 1 and block it. Okay, you need to know carbon monoxide and cyanide, which block complex 4. And you need to know oligomycin, which is a type of antibiotic that blocks ATP synthase, but it's not really used. Like, I'll give it to someone. I use this. It's only used in clinical research to see like like oxidative phosphorylation and what happens there. So rotenone, cyanide, carbon monoxide, oligomycin, but we'll focus on one mainly, the most popular, which is cyanide. It affects what? Complex 4. What's another name for complex 4? Cytochrome C oxidase. Going there. Okay, cyanide toxicity from nitroprusside. Nitroprusside is a drug that is used for people that are hypersensitive, high blood pressure. Now, when that drug is being metabolized, a byproduct of that is the cyanide ions. Big problem, because these cyanide ions, okay, they are byproducts of that. They will go and bind to the complex 4 in the mitochondria and the cells, right? And what will inhibit? It will inhibit electron transport chain. Inhibiting electron transport chain, you'll inhibit aerobic respiration as a whole. Cell will go into emergency mode, panic mode. Okay, I can't do aerobic respiration. I'll go to anaerobic respiration. And rather than part of it becoming acetyl-CoA, it will become lactate. Becoming lactate, lactic acid. Lots of acid, acidosis. Okay, so this is the steps. It's just a story. You have emergency modes, you have emergency pathways, all of these. Now, another cause of exposure to cyanide is not only this, fires, couch fires, fires as a whole. If you see a question about fires, it's probably asking about cyanide, but mainly couch fires will have cyanide fumes, which can be dangerous and cause all of this. Now, what are the symptoms? Now, because you don't have aerobic respiration, you'll have less ATP. Your cells will become lazy. You'll become lazy. Headache, dizziness, agitation, dyspnea. Okay. Nausea, vomiting, diaphoresis. It's because there is a difference in the pH in your blood. There's acidosis because of anaerobic glycolysis. A lot of anaerobic glycolysis are lactic acid, acidosis. Sweet almond odor on breath. That's because cyanide, from what I've heard, I think I've read sometime that cyanide is actually the molecule in almonds that gives its odor. I'm not sure, not 100%, but just know sweet almond breath from someone, it's not a good perfume. That means you have cyanide poisoning. Call 911. Treatments. You either have nitrites, which will convert hemoglobin to methemoglobin. What does methemoglobin do? It will take cyanide, and rather than binding to complex 4, it will bind to cyanide, inhibiting it from binding to complex 4. Or oxygen therapies, because it's not like, but this is only a temporary solution. When we have lots of oxygen, high concentration of oxygen, some of these electrons that, even though they're inhibited to be pumped in complex 4, the oxygen may be able to catch one of those, some of the protons, and produce water and have some NAD be recycled. But it's only temporary solution, and it's not like a permanent solution that will fully treat the guy. Now, okay, we said that there are inhibitors, but what about something called uncouplers? Now, all the electron transport chain. But what about if H plus, rather than going through ATP synthase, it has another pathway? Woo! Everything will happen. Electron transport chain will happen. NAD will be recycled. Oxygen will be consumed. But it's just H plus now will not move through ATP synthase. The main idea of producing ATP, it will not happen. Everything will happen, but just the most important step of ATP synthesizing, it will not happen, and the H plus will move through another pathway if they have another pathway. So NADH consumption, oxidation will be normal. Oxygen consumption will be normal, but ATP production will be down. Now you'll tell me, okay, the kinetic energy of these protons, okay, if they don't move through the ATP, then their energy, how is it utilized? What will happen? It will become heat. We even through moving through the ATP synthase, 50% of the energy is heat, and the other 50% is through ATP, production of ATP. But now, if it doesn't move to the ATP, the other 50% will become heat, and exclusively everything will become heat. And what causes H plus to not move through 80% synthase? It's molecule uncouplers. They just give an alternative pathway for H plus to move back into the matrix rather than going to ATP synthase. Now let's see examples. I'll explain this UCP protein. You'll have DMP, which was a drug that we thought that it treated weight loss. You have high doses of aspirin, will have some partial partial uncoupling and oxidative phosphorylation. And then you have one of the most important one, UCP1, a protein that will be found in your brown adipose tissue. You don't have brown adipose tissues unless you're an infant or a hibernating bear, but it's normal that can be found in infants and hibernating bear. DMP, actually, that drug, it was cancelled because people that took it, they were so hyperthermic that they needed to put them in ice because their body temperature was over the roof. Okay, but what about thermogen and UCP? Okay, brown adipose tissue. You told now, Alhassan, you tell me that it's in infants and bears. Why? Why it's not like in me? Because me and you are big creatures. We are 60 kilograms, 70 kilograms, even 80, pushing 100. We have large metabolism, and that metabolism will provide heat for our body temperature to stay the same, 37 Celsius. But for example, a baby, it's 2.5 kilograms to 3 kilograms. They need something to produce heat for them because of their metabolism is not enough. And that's where brown adipose tissue comes. Same thing with hibernating animals. For example, bears, they stay hibernating and sleeping for 2 to 3 months. I wish. I wish. لا حول ولا قوة إلا بالله. But okay, if they're hibernating, sleeping, their metabolism will also be low. So, oh my God, their temperature, they're going to become hypothermic. No, that's where their brown adipose tissue comes to play and just produce pure heat. How does that happen? Through the proton that they have in their mitochondria. The brown adipose tissues mitochondria have a proton called thermogenin or UCP1. It's basically telling the H plus, actually, you can go through me. And the ATP center is like, okay, it's seeing the electrochemical gradient, the H plus is finally coming. Whew! The UCP comes in clutch and takes them away and lets the H plus move through it. And it will not produce any ATP, rather, exclusively most of the energy will just be heat and no ATP production, nothing, and just pure heat that would be used to heat up the body for people that have, like if they have low metabolism, for example, infants or hibernating animals. Okay, now it's just the questions. Hopefully this recording works. Hopefully. Oh. You have a firefighter was rushed to the emergency room after inhaling the fire. What we said about fire, this is actually a past paper question, okay? Okay, fire, that means cyanide. Which of the following will be affected? Cyanide, it affects what? Oh, it affects complex 4. What's another name for complex 4? Cytochrome oxidase, cytochrome C oxidase. That's an easy question. Now, you will answer this. You can pause the video. Okay, now I'll answer. Which of the following can produce heat in the brown tissues? Brown tissue, same as brown adipose tissue of newborn babies. What is that special protein that brown tissue have? Oh, it's uncoupler protein, UCP1, thermogenin, the same name. And that's it, guys. Inshallah, you have a good TBL, and hopefully that recording works. 2 recordings before then, just I don't know. Hopefully this recording works, and Assalamu alaikum wa rahmatullah.